Good day, everyone. It's nice to see everyone again, and thank you for joining us today in our webinar exclusive. Exactly 123 years ago today, a British doctor named Sir Ronald Ross discovered that the female mosquitoes are responsible for transmitting malaria between humans. After his discovery, World Mosquito Day is observed annually on the 20th of August to raise awareness about the threat of malaria and other diseases transmitted by the world's deadliest animal. Today, we are commemorating World Mosquito Day by having Dr. Sebastian Horstmann explain to us what is insecticide resistance, how does it happen in mosquitoes, what are the different tools available, and how could we plan our mosquito control strategy better. Before we begin, a bit of housekeeping information. The webinar is scheduled for 45 minutes in its entirety, and that includes 30 minutes of presentation and 15 minutes of Q&A. Participants who are with us today, your audio and video are disabled. If you have any questions, please type them in the Q&A chat box and we will respond to them after the presentation. There will be a survey at the end of the webinar. Please click back to timeline and the survey will pop up on your screen. And finally, a recording of the webinar will be available to all registered participants after the webinar ends. Our guest speaker today is Dr. Sebastian Horstmann. He works in the vector control laboratory for field testing in Monheim, Germany. He is a biologist by training, and he scrutinizes new active ingredients and formulations for insecticides and screens them for effectiveness of mosquitoes. And he always does it with a smile on his face, as you can see in this picture here. He has more than 10 years experience and is an expert in field of mosquitoes, resistance management, biology, genetics, and the list goes on. Without further delay, let's bring on Dr. Sebastian to begin our webinar today. Over to you, Sebastian.
Thank you, Sepiao. Good afternoon, everyone. It's a great honor for me to speak here today to you, and I'm happy to take you on a short trip through the topics of insecticide resistance, mode of action, and the power of combining active ingredients. I would like to step directly into the agenda for today's presentation. I will touch topics today about what is insecticide resistance. I will talk about how does resistance develop in mosquitoes, then what types of insecticide resistance exist, and how we can tackle them. And then I focus on Bayer's approach of using active ingredient combination products. And I will further introduce two products, Fludora Fusion and Fludora Comax. At the end, I will, in general, talk about active ingredient combinations, and how we see this in terms of resistance development. After that, I'm glad to address your questions. Let's start with what is insecticide resistance. And of course, there is a general statement out there made by experts, here in this case, the Insecticide Resistance Action Committee, which is a group of experts, of industry experts, of companies that make R&D, have R&D activities. And they had to sit together and they said, insecticide resistance is a heritable change in the sensitivity of a pest population that is reflected in the repeated failure of a product to achieve the expected level of control when used according to the label recommendation for that pest species. A long sentence, and when people listen to that sentence the first time, they often have a question mark, like the person down there on the slide, have a question mark in their mind asking, what? And therefore, I made an additional slide. In short, you used to be able to control a pest population with a given insecticide, but now you can't. I think that's a little bit easier to understand. If we come to the point how resistance develops in mosquito populations, I have prepared a little animation. You should imagine now that you have a mosquito population, here visible by all these small individuals here. And there is one mosquito within this mosquito population that is, genetically a little bit different to the others. And these slight modification in the mosquito helps this mosquito to survive insecticide treatment. And now please imagine that you spray these mosquito population with a certain insecticide. Of course, a lot of these individuals die, but the red one and two others here in this example survive the treatment. Surviving means they can reproduce. And this is visualized here. Now your population is back and it contains more of the red ones. If you now spray your insecticidal product again, you can control this population, but some of them survive. The red ones again, the ones that have genetic, they have resistance mechanisms inside that helps to survive insecticidal treatment. Of course, they can reproduce again, and you have more of them. Now the mosquito population is in a state where the majority of mosquitoes is resistant. And when you spray your insecticide again, then you are not really able to control this population anymore, and they further reproduce. So the majority of mosquitoes is resistant now. Here is an overview slide, and you see the resistance development over time if you expose your insects to the same insecticide of the same mode of action over a lot of generations. At the end, your mosquito population is resistant. And we need to find ways to tackle this resistance. And there are different ways of how a mosquito can be resistant to an insecticidal product. I show you here on this slide the various mechanisms that exist and that are described as resistant. The first point is the behavior resistance. That's easily explained when you think about bed nets. When people sleep under a bed net, the mosquito cannot reach the host for a blood meal. Mosquitoes that tend to bite very late in the day, in a very late afternoon or beginning of the night, they probably cannot reach the host. They cannot bite the people sleeping under the bed net, and no biting means no blood meal and no reproduction, no egg laying, no reproduction. So these mosquitoes with a behavior to bite late at night, they cannot really reproduce. Others of the mosquito population that get up earlier and they bite in the early afternoon when people are not sleeping under a bed net, then they have more success in biting. They can take a blood meal, they can lay eggs, and they have offspring. Over the time, they tend to bite earlier and earlier the day and bypass more or less the bed net defense of the humans. And that leads to a change in behavior. The biting activity switch from late night to early afternoon. And that is what we describe as behavior resistance. The second point is reduced penetration. Maybe you know that in a lot of treatments, the mosquito lands on the surface and has to take up the insecticide via its tarsals. Now it can happen on a genetic basis that the cuticle or the wax layer on the legs of the mosquitoes are increased and they are thicker as usual. Now the uptake of active ingredients into the mosquito is reduced, and that's what we describe as reduced penetration. And of course, when less active ingredient enters the insect body, there is less activity at the targets, and therefore less effect of this insecticide. The third and the fourth point here are the most important resistant mechanisms, and I will later on focus very much on the next 2 points. The first is the metabolic resistance. The basis of metabolic resistance are enzymes that can degrade your active ingredient. When you think about having a keyhole and a key, and the key would normally fit to the keyhole, but the key enters the insect body and will cut into pieces. Then the key cannot reach the keyhole, which is in this case the target. So the enzymes are able to degrade your molecules before they can reach the target. That's the basis of metabolic resistance. Target site resistance means that the target itself has changed morphologically. If we come back to the keyhole and the key example, then the key can reach the keyhole, but it doesn't fit anymore because the keyhole has changed. This is what we describe as a target site resistance. All of these different mechanisms are a problem for itself, but of course, mosquitoes can combine these resistance mechanisms. They can have enzymes upregulated to have a metabolic resistance, and they can have a change in the target itself, a target site resistance, and these mosquitoes are called as multiple resistant. We have another term that we need to clarify, which is the cross resistance. When a mosquito has established, for example, a metabolic resistance to carbamates, just as an example, the chemical group of carbamates, and the enzymes can degrade the carbamate that comes into the insect, then you could think, now I apply an organophosphate, just as an example. But sometimes the enzymes can bind and degrade to the organophosphate as well. This is what we call the cross resistance. It is a resistance not particular against one chemical class, but to more. With this knowledge in your mind, we can go to the next slide and ask ourselves, what can we do to tackle resistance? I mean, we need to control the mosquitoes, and we need to find ways to deal with resistance and to be more effectful. And the World Health Organization, the IREC, and many other institutes have established several ways to work with resistance. I would like to start to describe here rotation on this slide. Rotation means that in one season you spray insecticide A, and in the next season you spray insecticide B. That means all the mosquitoes that survive treatment of insecticide A will most probably die in the next season when you apply insecticide B. That is the principle about rotation. Of course, you can enlarge this. You can spray insecticide C in the next season. And yeah, the strategy is to bring the mosquitoes in contact with a lot of different insecticides with a different mode of action. The same principle is mosaic spraying. Here you can imagine I have in a village, I have different houses and buildings, and I spray insecticide A in building 1 and spray insecticide B in building 2. Also here, the probability that a mosquito come in contact with different insecticides and different mode of actions is quite high, and that's the strategy in mosaic spraying. You can, of course, as well combine intervention methods. Here on the example, you see that people sleep under a bed net. The bed net is treated with a certain insecticide, and you have on the walls a wall spray with another insecticide with different mode of action. So when a mosquito enters here the building, then it will come in contact with the insecticide on the bed net, and probably as well with the insecticide on the wall. This is the strategy about combination of interventions. All these three mechanisms that I have described have one one disadvantage. The mosquito need to deal only with one insecticide per time. In rotation, it is only one insecticide in one season. In mosaic spraying, it is in building one only one insecticide where the mosquito has to deal with. And in the combination of interventions, the same. When the mosquito is on the bed net, it's one insecticide. Then the mosquito flies to the wall, it's another insecticide. It is, of course, a challenge for the mosquitoes, but at the same time, they have only to tackle one insecticide. This is different to insecticide combinations. Here you have two insecticides with different mode of actions in one product. That means in the moment when the mosquito come in contact with this product, it need to tackle or it need to deal with two insecticides at the same time. And that is the big hurdle for the mosquito where we think we can benefit from. We are not the only ones at Bayer. The World Health Organization and also the IREC committee recommend mixtures. Here's a statement of the World Health Organization from the so-called GPERM document. That is a documentation about how can we tackle resistance in future. And they explain here that mixtures or combination products guarantee that the mosquito come in contact with two modes of action at the same time, and that they are not available yet, but they will be the future in malaria control and in insecticide resistant management strategies. The GPERM document was published in 2011, and today we can say we have combination products on the market. I introduce here the first one, which is Fluidora Fusion. Fluidora Fusion is a product made for indoor residual spraying. That means you spray the indoor walls of your buildings, and it contains two active ingredients, clothianidin and deltamethrin. Clothianidin belongs to the IREC mode of action class 4A. It is a neonicotinoid, and I will come to the mode of action later in this presentation. The other active ingredient is deltamethrin, which is, as you probably know already, a pyrethroid belonging to a different mode of action class, the class 3A. This product, Fluidora Fusion, is PQ listed. That means that the World Health Organization has checked this product and they recommend it especially for malaria control. For a spray that you bring onto inside walls, you need a long residual activity because you cannot spray it every day or every week. It's a lot of effort, so it needs to last as long as possible. And Fluidora Fusion was intensively tested in countries all over the world, and we can say they have a confirmed efficacy of 6 to 8 months or beyond, depending on the surface material. You can imagine that concrete with a high pH value is more aggressive to the product as, for example, a glazed tile. And therefore, the residual effect is different. But with 6 to 8 months or even beyond, Fluidora Fusion is a really long-lasting IRS product. I would like to go into a little bit more detail of IRS. Indoor residual spraying means spraying the inside walls. And this is one of the key methods in malaria control. You see here on the picture on the left side, one sprayman spraying the inside walls of huts. This method is WHO, World Health Organization, recommended, and it targets mainly the malaria vector, which is Anopheles. But nowadays, a lot of research institutes figured out that it's probably also a good method to control Aedes mosquitoes. I have a publication shown there that has studied the effects of indoor residual spraying against Aedes aegypti mosquitoes. And they see that Aedes mosquitoes rest on certain places. And if you treat these places, then you have an effect also in controlling Aedes mosquitoes. That's what they call the targeted indoor residual spraying. Now we come to the point, what is Fludora Fusion doing when a mosquito come in contact and take up the two active ingredients, clothianidin and deltamethrin? What you see here on the right side is the connection between two nerve cells. Nerve cells in the body of everyone in the world need to communicate with each other. And they do it inside of a nerve cell with an electric impulse. But they have also connections to other nerve cells. And here they need a chemical way to transmit the information of an impulse. And they do it in this gap region. You see the presynaptic cell, this is one nerve cell, and you see the postsynaptic cell on the other side. And in between, you have the presynaptic, or you have the synaptic gap. When the nerve cells want to communicate with each other, a transmitter called the acetylcholine into the synaptic gap. On the other side of the cell, on the postsynaptic cell, you have acetylcholine receptors, and they bind the acetylcholine, open so-called sodium channels, and that leads to a sodium influx, and the impulse will be created in this next nerve cell as well. Then you have acetylcholine esterase enzymes that cut the acetylcholine into choline and acetic acid, and they take up and recycle these molecule parts. If you bring clothianidin into the system, then clothianidin mimics the acetylcholine. It binds to the acetylcholine receptors and trigger the sodium influx by opening the sodium channels. The problem for the organism here is that the acetylcholine esterase enzymes, they cannot degrade clothianidin. So it stays in this presynaptic gap and pushes and triggers all the time the acetylcholine receptors. Deltamethrin is the counterpart of in this dual mode of action that we have in Fludora Fusion. It contains a type 2 pyrethroid, and this kind of pyrethroids, they like to jump into open sodium channels, and that's the benefit here, because clothianidin is opening sodium channels, and deltamethrin can use the open state of these sodium channels to be effective as well. That is what we describe as complementary activity. The two active ingredients complement each other. This is a lot of theory, of course, and we want to see what is the effect, the complementary effect in the field. And therefore, we have tested Fludora Fusion in a lot of countries all over the world. And you see here an example from the Ivory Coast. Here, the product was sprayed on mud surfaces. Mud is a very challenging surface because it contains a lot of different ingredients, and it has also pores and cracks and crevices where the product can trickle away. We tested here in this trial not only Fludora Fusion, we tested the single active ingredients, deltamethrin and clothianidin as well, because we wanted to see, is there really something additional, or is it just one active ingredient that we see here? And if you look on the green bars, which is deltamethrin, then you see that 1 month after treatment, shown on the x-axis, MAT means 1 month after treatment, then you cannot control 100% of the test mosquitoes. You are already below 100%, but you are above the 80% threshold. So you can say deltamethrin is working 1 month after treatment. In the second month, the efficacy touches the 80% threshold already. But it's above the threshold, or at the threshold, you can say there is convincing activity of deltamethrin. After 3 months, not anymore. Now you can up to 40 or 30% of the mosquitoes So this is not a real activity anymore. So deltamethrin as a single product lasts here, in this case, only 2 months. If we look on clothianidine, shown here with the pink bars, you see it's 1 month more of efficacy. But after 4 months, it also drops in efficacy. When we now look on fluorofusion with the blue bars, then you see it lasts up to 6 months. It is 6 months above the threshold of 80% control. And that means the combination of the two active ingredients lasts double as long as clothianidine alone, and it triples the effect of deltamethrin. And this is against pyrethroid-resistant mosquitoes. So what we describe as complementary activity is also visible in field trials. There is another product on the market that combines active ingredient, or will come to the market, sorry, I'm a bit too early now. It's not yet available, but it will be in the future. It contains flupyridiferone and transfluthrin. Flupyridiferone is not a neonicotinoid. It belongs to the butanolides, but it has the same mode of action. Transfluthrin is also a pyrethroid. So we mix here, again, the modes of action that I have described before. Basically, fluorofusion is not made for indoor residual spraying. It is a space spray, which is probably very well known in your respective country. When you see dengue emergency cases or raising cases of dengue somewhere, you very often send spray operators into that region to push down quickly the virus-carrying mosquitoes. And we have developed this product here for exactly this use, space spraying. We have submitted it for WHO for testing, and the PQ process, the PQ recommendation process, is still ongoing. This combination of the two active ingredients provide a very fast knockdown and an effect for 24 hours. In this product, it's not necessary and not desired that it lasts very long. It's not for a long-term efficacy. It is an immediate action to push down the mosquito population as fast as you can. Here you see the different ways of how this space spray can be applied. On the left side, you see the truck-mounted fogging machines or aerosol generators. And on the picture on the right side, you see handheld devices to create a cloud, an insecticidal cloud of tiny, tiny droplets. Every mosquito that flies into the cloud will be pushed down and cannot bite anymore. The main targets here are Aedes mosquitoes and Culex mosquitoes, but there are tests ongoing in some research institutes to address this for Anopheles control as well. If we go to the mode of action, then we have more or less the same situation like for fluorofusion. Here, the flupyridiferone mimics the acetylcholine and opens the sodium channel, and transfluthrin targets the open sodium channels. So this is probably, this is more or less the same way of acting like in fluorofusion. But here we choose other active ingredients, and that has also good reasons. Flupyridiferone is a new active ingredient which was never used in vector control before. We see here a benefit, especially to tackle insecticide-resistant mosquitoes. Not only that, flupyridiferone is, we call it, inspired by nature, because it's the basis of this compound was a natural compound called stemophyllene, which is part of the plants Stemona japonicum, which are probably known especially in the Asian region. The flupyridiferone was the one active ingredient we choose for this product, but we choose transfluthrin as well, and that has also a very good reason. because transfluthrin is a special pyrethroid. You see now on this slide the way how a mosquito degrades pyrethroids. And on the left side, you see the publication, which come from the UK, from the Liverpool School of Tropical Medicine. They have identified the way how a mosquito cuts a pyrethroid into pieces, that it's not effective anymore, what I have described as metabolic resistance. You see the red arrow, that is the target point for the enzyme. On this side, on this double ring structure, the enzyme has to bind. And if the enzyme can bind, it will hydroxylize the molecule. It puts the OH group onto this outer ring. That is in degradation. The next step is to cut off this outer ring structure on this pyrethroid. In this example, it's deltamethrin, but you will see when you compare all the molecules of pyrethroids that are on the market, this double ring structure is present also in cypermethrin, in permethrin, in all these common pyrethroids. If we look on the transfluthrin, then it's different. Transfluthrin has only one ring, and this ring is fluorinated. And that makes it impossible for the enzyme to bind there. And when the enzyme cannot bind there, it cannot make the reaction step, so it cannot degrade transfluthrin. And this is also published in the publication on the lower right side, these findings. And that transfluthrin provides a kind of resistance breaking potential for this way of degradation. To make it a little bit more visible for you and easier to understand, I made a drawing here. In red, you can see the deltamethrin, and you will see on the next slide how it is degraded. And on the right side in yellow, you see transfluthrin. If we now have deltamethrin here, and you have two enzymes in the mosquito that want to degrade, oh, that was a bit too fast, sorry, and they want to degrade the molecule, then you see this is the first degradation step, and there is a second one to cut the pyrethroid into pieces. If the same enzymes try to do it with transfluthrin, it will not work, because transfluthrin has its outer ring protected. At the end of my presentation, I would like to come back to the mode of actions that I have described before, and why we see combination active ingredients in a product as the advantage for controlling mosquitoes. I listed in the blue windows on the left side, I listed what is necessary for the mosquito to survive treatment. And here I focus on target site resistance and metabolic resistance. If the mosquito has a target site mutation at the acetylcholine receptor, then it can withstand probably clothianidin or flupyridiferone. That would be a benefit for the mosquito. If I have on top a target site mutation at the sodium channel, then this inhibits the pyrethroid and is probably a benefit to survive treatment of deltamethrin or transfluthrin. On top of this, I need detoxifying enzymes for clothianidin and deltamethrin at the same time. And I need enzymes, if I focus here on COMEX, for flupyridiferone and transfluthrin. And of course, I can combine these target site resistance with the metabolic resistance, what I described as multiple resistance. But this is a long way to go and a very high hurdle for the mosquito. It needs to establish target site mutations, not only on one target. I need it on two, on the sodium channels and on the acetylcholinesterase. And I need on top enzymes that can degrade clothianidin and deltamethrin at the same time, or flupyridiferone and transfluthrin at the same time. And that's a lot of work for the mosquito and a higher hurdle to survive in our view. If we compare this to a single AI product, and I used here clothianidine as an example, we just in brackets, we just need a target site mutation and on top probably detoxifying enzymes to survive treatment of clothianidine. Therefore, just by counting the different ways that a mosquito need to survive treatment, we see that in a combination product, we have a higher hurdle for the mosquito. And that's why we see combination products for the future and to tackle resistant mosquitoes. So I hope I was able to transmit a little bit of information how resistance develops, what resistance is, and what strategies are on the market to tackle resistance. I hopefully have described what is indoor residual spraying and space spraying and have introduced the products Fludora Fusion and Fludora Comax. We see, just to end this presentation, we see the combination products as the future products in mosquito control because they provide a big challenge for the mosquito to survive. And with this, I would like to end my presentation, and I'm very happy and glad to answer your question about insecticide resistance, mode of action, mosquito control, or whatever you want. Thank you. And I hand back now to Sepiao.
Thank you, Sebastian, for an extremely educational session. Now let's move to our Q&A session and see what questions we have received so far. Right. Are you sure you want to answer any questions? Right. Here we go. Is there a way to determine whether mosquito population I'm trying to control is resistant to the product I'm using?
Yes, there are a lot of so-called susceptibility assays on the market. You probably have heard about the WHO tube assay, which uses insecticide-treated paper material. On top of that, you have the so-called CDC bottle bioassay, where you bring your active ingredient into the inside wall of a glass bottle. You put mosquitoes inside, and then you see if they can be controlled or not. So there are ways to check the resistance status in a bioassay. Of course, research institutes have more possibilities. They can check these mosquitoes also on a genetic basis. They can directly see, is there a target site resistance? Is there increased enzyme levels somewhere? They have more possibilities, but yes, this is checkable as well.
Okay. All right. Second question, how do I know which kind of resistance, for example, behavioral resistance, reduced penetration, metabolic resistance, target site resistance, how do I know which one the insects I'm trying to control has?
Yeah, this is what I said. When the institutes have the possibility to check the genes or make a so-called transcriptome analysis with the mosquitoes, then they can directly say that enzyme levels are upregulated. That does not necessarily say that this enzyme is involved in the insecticide degradation, but at least they can see these enzymes are upregulated, and there is already a lot of knowledge in available what enzyme groups are involved in insecticide degradation. So there are ways to check this, and yeah, we are doing this as well, by the way.
Okay. Okay.
Yeah, we want to characterize all our mosquitoes.
Yeah, absolutely. Next one, we have how long does it take, usually take for a population of mosquitoes to develop resistance? And how long does that resistance last? Does it last forever?
in worst case, yes. It depends on the selection pressure. As I have shown in the video, when you apply the same insecticide again and again, then you select for mosquitoes that have a resistance mechanism, and as long as you bring them into contact with the insecticide, there's no reason for them to skip these resistance mechanisms. So if you reduce the selection pressure for the mosquitoes by not applying an insecticide, which is also not a solution, but if you apply a different insecticide, then you can probably push down the resistance mechanism to the previous one. Your other question was the resistance development. This is very different and depends very much on the mosquito strain you work with. Some mosquitoes have a fast tendency to develop resistance mechanisms. Others are less exposed, or they are not so much come in contact with the insecticide, and then it takes a little bit longer. So this can be very, very different. And the same is with the long lasting of resistance mechanisms. Some are not creating fitness costs, and then mosquitoes can perfectly live with it. Some others have disadvantages for the mosquitoes, and then when you don't have the selection pressure anymore, then the mosquitoes tend to return to the wild type, the non-resistance mechanisms.
Okay. Next, we have the question on this. If the mosquito is resistant to pyrethroid and one of the AI in my product is a pyrethroid, will it still work on the mosquito?
Yes, in a combination product, you need to see both mode of actions because the mosquito has contact to both mode of actions at the same time. Now, you could say the one mode of action is not working, but the other. But in our products, we have seen this, what I try to describe as complementary activity. We saw that even if you have pyrethroid-resistant mosquitoes, you see an increased efficacy against these mosquito strains. And this is, or I try to explain this by this complementary mode of action, this complementary activity in the products.
Okay. Next, we have, will using a dual mode of action combination product reduce my options of product to rotate in my program?
No, I don't think so, because what you need to rotate are mode of actions. And as I said before, in a combination product, I need to see both mode of actions as the effective issue in this product. That means I can perfectly use it in rotation strategies. When I have, for example, used a pyrethroid in one season, and I now would like to use Fludora Fusion, it's also possible because pyrethroid mode of action is only one part of Fludora Fusion. The other part, or the complementary effect, will control your mosquitoes. So even if you have used pyrethroids before, they will not survive treatment of Fludora Fusion.
Right. Okay, that makes sense. What is the best way to integrate chemical intervention with non-chemical tools like Wolbachia? We see a lot of countries running trials on Wolbachia these days.
Yeah, and yeah, that's very interesting and a very great approach. And yeah, I see that we probably can combine the two techniques. If I take the example with the Wolbachia, you release mosquitoes into a certain area, and I think there are ratios, like I need to release what was it, 10 mosquitoes, 10 male mosquitoes for one female wild-type mosquito. So you need to produce a lot of Wolbachia-carrying mosquitoes. And I think if you use a spray spray before, and you can push down the mosquito population, you have probably a better effect afterwards in releasing the Wolbachia-carrying mosquitoes. So I think there are strategies and ways to combine chemical effects and biological effects.
Right. Okay. Let's see what we have next. Does Bayer have any upcoming new products to counter resistance? You've just presented those two products.
Yes, I would.
Yeah.
I would say Fluidora Fusion is a good one, and Fluidora Comax is also nice.
For sure. Is mosquito resistance a more prevalent issue in certain countries, you think? What do you think?
Oh, that's a good question. Yeah. Some publications or some people say that you have resistance always where you have entomologists. So in some areas, you simply don't know really what's in the mosquito population. And in other countries, you have a very good research institute. They check all the mosquitoes flying around. And then it tends to look like in this country, I have more resistance than in the other countries. But you need to keep in mind always, is it really checked there? And yeah, I would recommend a strategy to check the mosquitoes in a way that we need to know what's in the mosquitoes, what type of resistance, and then we can make tailor-made products to tackle these kind of mosquitoes.
Yeah. This is interesting. Are mosquitoes more likely to develop resistance to generic products?
I think this is not the case. It depends very much on the active ingredient. I mean, when you have a new active ingredient and you know that the R&D companies they do research on new active ingredients with new modes of action. And of course, we think about when we have a new mode of action never used in vector control before, it will be able to tackle resistance quite well. Of course, that need to be checked in field trials because you can have this issue of cross resistance. Yeah, but these new active ingredients, they have, from my point of view, a benefit in controlling the resistant mosquitoes. Yeah, but I think it depends very much on the active ingredient.
Okay.
You cannot say it's a generic, so you should use another one. That's not the case, no.
Yeah. In your opinion, is there one type of resistance that is more difficult to overcome than others?
Ooh, also a very good question. I think
we have a tough crowd.
Yeah, very good, very good. I see the metabolic resistance as a very challenging type of resistance because the enzymes are very active. They are very fast in degrading the active ingredients, and they can probably degrade not only one active ingredient but several ones. So if we have increased enzyme levels in mosquitoes, this is really difficult to overcome. Target site mutations as well, target site resistance as well. If the target has changed and the insecticides cannot bind really anymore, then we can hope that it causes fitness costs in the mosquitoes, but it's not a given. So target site resistance and metabolic resistance are very, very tough ones and the most common ones as well. If we have reduced penetration, I would say we might have a way to change the uptake. You know probably that some active ingredients are more volatile than others. So when we see there is an increased cuticle thickness and the penetration is reduced, then we probably can do it via the respiratory system of the mosquitoes. So here I see more chances to work with. And behavior resistance, okay, then we need to be more clever and find also, or make also behavior changes in us.
Okay. Right, next we have this. I believe Feudora fusion is effective on KDR strains of Anopheles species. Am I correct? Also, how do you see the possibility of managing insecticide resistance combining limited technologies such as residual technologies and using AIs for longer time?
Yes, first, the KDR resistant strains can be controlled with Feudora fusion. That we have tested it with more than 11 different strains, and we tested it with different KDRs, the East African KDR, the West African KDR. So that is possible, and yeah, if you're absolutely right concerning your strategies. We have very limited active ingredients available, and therefore we need to find strategies to keep them effective for the future. And we at Bayer, we see combination products as one way to keep the active ingredients effective for a long period of time, because the mosquito cannot focus on just one new active ingredient. Clothianidin, for example, is very new as well in the vector control market. And if you have just one active ingredient, then the mosquitoes can focus on this, and evolution can focus on this and can build up resistance mechanisms. That can happen in a combination product as well. Don't get me wrong, this evolution, nature finds a way, but we see it as more difficult if the mosquito or if the evolution process need to deal with two attack points, with two active ingredients and two modes of actions at the same time.
Okay. One question here. Is there any published data on the impact of rotation spraying on malaria burden? Could you elaborate?
I know there are papers out there, but I don't have them in mind yet. I would need to check. But
okay.
Probably you have the email address of this question, then I can share the publications. They are existing, yeah.
Okay, sure. Next one we have, do we know the magnitude or importance of resistance mechanism reduce penetration? So let me just repeat this. Do we know the magnitude / importance of the resistance mechanism reduce penetration? So this is one of the resistance. Okay. And at least in plant protection, there are penetration enhancer used. So would it make sense for us to use similar chemistry in vector control products?
Absolutely, it makes sense, and Bayer is doing research on this field, but the research, I mean, we have other ways of treatment. If you treat a wall, for example, a porous wall, and you have uptake enhancers, then you increase the uptake into the wall as well. And then it's probably not available on the surface. Everything is in the wall. So you need to find clever strategies to combine penetration enhancers with active ingredients. But we are working on this, and there is at the moment a CDC bottle assay in the public domain for testing, and that there is a modification in this CDC bottle assay to make it usable for neonicotinoids as well. Neonicotinoids tend to crystallize very strongly, and then the mosquitoes have no uptake anymore. And in this CDC bottle assay, on a glass bottle, exactly this happens, and therefore we have modified this CDC bottle assay with a penetration enhancer.
Okay. In an outdoor setup in a tropical country, for example, when we have rain and sun in the Philippines in the tropics, how long will this chemical last? And I'm assuming this is through the fusion in terms of residuality. 1 month, 2 months?
I would expect longer. Sorry, I need to make one step here. You should not forget that you spray the Fluidora Fusion on the inside walls. So direct sunlight is probably not that big issue. Humidity, of course, and therefore we have tested Fluidora Fusion in tropical countries very, very intensively. And we see in countries of Africa where you have very high humidity and very high temperatures, depending on the surface, efficacy for up to 12 months. And therefore, I expect that you will have more than 2 months on your surface.
Yeah, I believe so.
I
what's
believe
the
very
field?
much, yeah.
Yeah, yeah. What's the field trial protocol for testing a new product that offers a combination mode of action?
And they are very similar to a standard IRS products. It is just the difference that with the treatment you apply not one mode of action or one insecticide, you apply two, but from the technique itself, it's very similar. And the same for the spray spraying. It makes no difference. It's also a product in a bottle or in a sachet, and you can mix it very easily in your backpack sprayer or your aerosol generator and apply it like every other product as well.
Okay. Let's just take a couple more questions. Dr. Horstmann, as you mentioned cross-resistance a while ago with the product with combination AI, will it lead to cross-resistance effect after several uses?
Yeah, we cannot really say that now, but from a scientific point of view, as I said, nature will find a way. Evolution is a process that we will not stop. What we do is we try to slow down this process, and therefore we create higher hurdles for the mosquitoes to overcome. But if you would use such product again and again over a long period of time, then you will select for so-called generalists. These are then mosquitoes combining a lot of resistance mechanisms, and in worst case, they can overcome the treatment of a combination product as well. This might happen.
Okay. Right. I think that's all the time we have for today. I think we only have a couple of minutes left. But before I let you go, Sebastian, would you like to say a few words to our audience?
Yes, I'm very happy that I had the chance to speak to you here today. I'm very grateful that you took your time, and I hope I was able to transmit a little bit more knowledge about resistance, about our products. And I hope to see you next time in probably another webinar or on conferences as well. Hoping that we can travel again in the near future.
Yeah.
Thank you.
Same here. Thank you, Sebastian. Stay safe and have a good rest of the day. Okay. That brings us to the end of our webinar today. Thank you so much again for your active participation. We apologize for not being able to answer all your questions, but if you could leave your details together with your questions to the email address that you see on the screen, we will get back to you as soon as we can. So thank you once again. Stay safe, and we'll see you next time. Goodbye.